با درود و سلام خدمت همه شما بینندگان عزیز تلویزیون جهانی کانال 1. من دکتر نگار متیقنی با تازه ترین یافته های پزشکی خدمت شما هستم. اول میریم سراغ اخبار کوید و تازه ترین اطلاعات و آمار در مورد این پاندمیک. طبق اسلاید اول لطفاً اتاق فرمان، طبق آخرین یافته ها توتال کانفرمد کیس ما 196.6 میلیون گزارش شده، توتال دث 4.1 میلیون بوده. از این تعداد کیس های جدید 657.714 بوده که تعداد مرگ ها به 11,644 رسیده. اسلاید بعدی لطفاً. از لحاظ واکسیناسیون در کشورهای مختلف و در دنیا per 100 people به ازای هر 100 نفر مردم واکسن، آمار جهانی 51.5 هست. این آمار برای کشور چین 111، برای کشور هندوستان 32.7، برای ایالات متحده آمریکا 102.7 و برای آلمان 108.6، کشور انگلستان 126.5، ژاپن 65.3 و کشور ترکیه 83.5 گزارش شده. اسلاید بعدی لطفاً. این آمار برای کشور روسیه 40.6 در حالی که کشور کانادا 128.6 و برای کشور کره جنوبی 47.3 هست. اسلاید بعدی لطفاً. این آمار برای امارات متحده عربی 168.3، برای کشور استرالیا 45.5، برای کشور اسرائیل 128.5 و برای کشور ایران 12.1 گزارش شده. اسلاید بعدی لطفاً. همونطور که توی برنامه‌های قبلی هم دیدیم، در این نمودار کشورها با بالاترین تعداد کیس در قاره‌ها و کشورهای مختلف نمایش داده شده. در قاره اروپا کشور روسیه در هفته‌های قبل بالاترین تعداد کیس رو داشت که در هفته‌های اخیر کشور انگلستان در حال حاضر بالاترین آمار کیس رو داره. بعد از انگلستان اسپانیا، روسیه و فرانسه قرار گرفتن. اسلاید بعدی لطفاً. در کشورهای آمریکای لاتین برزیل بالاترین آمار رو داره. بعد از برزیل مکزیک، آرژانتین و کلمبیا قرار گرفتن. اسلاید بعدی لطفاً. در میدلیست یا خاور میانه ایران همچنان بالاترین آمار کیس رو داره. بعد از ایران عراق، اسرائیل و امارات متحده عربی قرار دارن. در ایران در حال حاضر گفته میشه دیگه پیک پنجمی صحبتش نیست. در حال حاضر به صورت دراز مدت پیش بینی شده که وضعیت به وخامت خواهد گرایید. مقدار سرومی که توی بیمارستان ها هست ناکافیه. اکثر شهرها به عنوان قرمز اعلام شدن و برای شهر مشهد حتی وضعیت سیاه گزارش شده به علت آمار بالای مرگ و میر. حتی یکی از مقامات بهداشتی گزارش کرده که آمار مرگ و میر روزانه به بیش از 600 تا 800 نفر در روز خواهد رسید و در حال حاضر گفته شده که هر 4 دقیقه یک ایرانی متاسفانه جون خودش رو به دلیل این بیماری از دست میده. اسلاید بعدی لطفاً. یک تحقیق جدیدی در مجله نیچر چاپ شده که چطور کرونا ویروس سلول ها رو مبتلا میکنه و چرا دلتا اینقدر خطرناک هست. این یک تحقیق خیلی طولانیه ولی یک سری مطالبی به صورت خلاصه مشخص شده که ظاهراً بعد از ورود به سلول این ویروس میتونه تغییر شکل بده و حتی یک پوشش از ترکیبات قندی مثل گلایکن دور خودش داشته باشه که شوگر کوتد میکنه خودش رو و حتی تشبیه کردن به گرگی که لباس گوسفند تنش کرده و میتونه از دسترسی ایمنی فرار بکنه و فرمانهای مختلفی رو برای سلول صادر بکنه. اسلاید بعدی لطفاً. یک تحقیق دیگه‌ای که توی نیچر بهش اشاره شده اینه که تحقیقات روی آنتیبادی ها، نیوترالایزینگ آنتیبادی ها به شدت ادامه داره، ببینن اینها به مرور زمان از بین میرن یا نمیرن و میان بررسی میکنن که چه کسی میزان آنتیبادی کمتر یا بیشتری داره و میزان و تیتر این آنتیبادی ها در آینده نزدیک تعیین کننده خیلی مهمی در ابتلا به بیماری و واکسیناسیون خواهد داشت. اسلاید بعدی لطفاً. همینطور در مجله معتبر پزشکی نیچر در مورد کوید بوستر واکسین صحبت شده که یک سری کشورها که از موقعیت بهتری برای واکسیناسیون برخوردارند در حال حاضر این رو وارد گایدلاین های خودشون کردن یا به صورت خیلی جدی در نظر دارن بکنن و یکی از این کشورها کشور اسرائیل هست که دوز سوم رو در هفته اخیر برای سالمندانش و برای کسانی که ضعف ایمنی دارن به عنوان دوز بوستر در نظر گرفته. ما مهمون برنامه‌مون امروز یک دانشمند بسیار برجسته هست از دانشگاه استنفورد در ایالت کالیفرنیا، دکتر بروس پاترسون. آقای دکتر پاترسون 4 اپیدمی رو گذروندن و توی این برنامه که ما خیلی خوششانس بودیم تونستیم ایشون رو داشته باشیم راجع به کووید تازه ترین اطلاعات رو خواهند داد در مورد بیمارایی که کووید میگیرن و بهشون لانگ هالرز گفته میشه، علائم رو بعد از بهبودی چندین ماه حس میکردن، ادامه داشتن این علائم و راجع به واکسن ها و اون چه که در حال حاضر جدیدترین اطلاعاتی که در مورد این بیماری موجود هست رو آقای دکتر پاترسون در اختیار ما خواهند داشت. این اینترویو به زبان انگلیسی هست، ما حتما ورد بای ورد این اینترویو رو در اسرع وقت در خدمت بینندگان عزیزمون خواهیم داشت. This is Dr. Negar Motayaqeni from Channel 1 TV. Today I am so honored to interview a rockstar scientist, a legend, a potential Nobel Prize candidate, Dr. Bruce Patterson from Stanford and Inseldex. Dr. Patterson, thank you so much for accepting my invitation. Thank you so much, sir. At the beginning, would you let our audiences know more about your background?
Sure. I'm a viral pathologist. I trained at Northwestern University Feinberg School of Medicine, and I did a postdoc in virology. This was all in the late 80s and early 90s at the time that HIV was just as big of a problem as COVID is now. So, you know, it was a time of a lot of research, a lot of new discoveries, and as I tell everybody, this is my 4th pandemic, so I'm getting used to it, unfortunately.
Thank you so much, Dr. Patterson.
Then I went on to direct virology at Stanford University Medical School, and then founded IncelDX in 2010, which is a molecular diagnostics company focused on multi-omic detection of genes and proteins at a single cell level, which with a focus in oncology and obviously in infectious diseases.
Thank you very much, Dr. Patterson. For the first question, I would dive into the long haulers. What is long hauler and who are long haulers?
That's a great question. When I was in the middle of doing trials for therapeutics in acute COVID, beginning in February of 2020, after a trip to China in January of 2020, we noticed at the end of these trials that, you know, people were feeling better, people were alive, but they were going home with their immune systems not even close to being normal. And I was struck by the fact that we had this population of people post-COVID that had profound immunologic abnormalities. And then they started having symptoms 3, 4, 5 months at the time post-COVID. We now know it's out to 16 months, maybe even going on 17 months now. And what we did was in the summer of 2020, we started to use machine learning and artificial intelligence to figure out what was going on in these individuals who had persistent symptoms long after they supposedly cleared the virus and recovered. And sure enough, we found a very distinct immunologic entity that was very, very different from acute COVID that is now called long COVID, long haulers, PASC, you name it. But we found and just published a couple of weeks ago, it came out in print, the results of our artificial intelligence and machine learning that showed that it is indeed its own condition. It's very real, but it's also very different than acute COVID.
Thank you very much. Why these flare-ups happens, Dr. Patterson?
Well, that was the big question. The question was why, and when I looked at the first immune profiles in the late summer of 2020 and early fall of 2020, I was struck by the fact that there was a distinct antiviral component that was even more appropriate than what you would see from the innate immune response in acute COVID. But the other thing that really struck us was the fact that markers of blood vessel inflammation were significantly elevated in these individuals. And at the time, we didn't know why. So we went on to look further, and just recently, and we just sent this paper in for peer review, we found that this certain class of white blood cells that travels all over the body called monocytes contain the S1 protein of COVID 16 months after infection, and in the total absence of intact RNA. So these cells don't have replication competent virus, yet still migrate around the body and cause inflammation due to the S1 protein.
Wonderful. Are these replicating RNAs or just debris?
No, the RNA is in fact fragmented, so it's debris. The S1 protein is being presented by these cells, which is one of their functions, is antigen presentation. The other function of these particular cells is that they patrol the blood vessels. And so this really put our two investigations together. The first paper, which showed that long COVID was caused by vascular inflammation, and then the second paper saying the vascular inflammation is being caused by the fact that these cells, which contain COVID protein 15 months after infection, are binding to the blood vessels through a pathway called fractalkine, and causing the blood vessel to become inflamed. And these cells, of course, cross the blood-brain barrier, so it can account for the brain fog and ringing in the ears and this head pressure that is described in a lot of long haulers. And the other thing is these cells are mobilized by exercise. When we looked in the exercise physiology literature, they're mobilized by exercise, which is why, to a person, long haulers have exercise intolerance, and that exercise actually makes them worse. There's so many clinics out there that are trying to treat long COVID with physical therapy and exercise, when in fact that's probably the worst thing that could be done.
Thank you. And my other question goes into the different news we are hearing about vaccination and side effects such as myocarditis, and even if recently Guillain-Barré. Would you please let us know more about these complications and side effects?
Yeah, I mean, again, you have to realize how many people have been vaccinated, and it would be naive to think that there weren't going to be side effects to a vaccine. We've now enrolled over 500 post-vaccination patients that have long hauler symptoms, despite the fact that they've never been infected by COVID. And we're finding a lot of similarities that we're about to send in a new paper with long haulers themselves in terms of the immunology. We took the immune profiles of those post-vaccination individuals and put them through the same machine learning artificial intelligence algorithms that we use to first identify long haulers, and remarkably, they look identical to long haulers, symptom-wise and immunologically wise. But I would say that they have less vascular inflammation, although they have some, than long haulers, and we've been very successful treating the post-vaccination long haulers, if you will, in the same vein that we treat long haulers themselves.
Great. Very few minutes ago, a collaborator colleague from Harvard informed me that they are collaborating with a research team in India. They have been evaluating patients who have recovered from COVID, and now they are undertaking elective surgeries. They have found out that they are more susceptible to coagulopathies. How do you define this problem, since we have seen this with other vaccinations like Johnson & Johnson?
Well, again, you have to go back to the pathogenesis of the virus itself. I mean, we see in acute COVID this vascular inflammation as well, which is what I think is somewhat unique to SARS-CoV-2. And, you know, when you have vascular inflammation, obviously attracts platelets and other molecules involved in the coagulation pathway. And of course, platelets produce a protein called soluble CD40 ligand, which we find elevated in long haulers and acute COVID. And that is produced by activated platelets. What activated platelets also produce is a protein called RANTES. RANTES attracts other immune cells. And so you have this vicious cycle of vascular inflammation, which brings in other immune cells, which produce other pro-inflammatory cytokines, which increase the amount of inflammation. And you have a setup for coagulopathy. And so, you know, the approach should be two-pronged. Number one, decrease the inflammation, and number two, obviously prevent coagulopathy. So commonly we'll put individuals with acute COVID and even long haulers on, you know, baby aspirin to, you know, prevent any of this clotting.
Thank you so much. Dr. Patterson, do you envision annual COVID boosters along the lines of flu shots? If so, would you be worried about unforeseen consequences in terms of the overall effectiveness of immune surveillance in an era of increasing reliance on vaccine?
Well, again, I think we're in a battle with the virus. And we saw the same thing in HIV. We're developing drugs, we're developing vaccines, we're developing approaches to target this virus, and the virus is fighting back. And the virus fights back by mutating. And HIV was probably had one of the highest mutation rates going. Hepatitis C has a high mutation rate. There's never been neutralizing antibody or vaccine for hepatitis C. COVID, obviously there's vaccines, but the viruses, unless you completely eliminate replication throughout the world, the virus will continue to mutate, and we'll see new variants popping up. And we'll have to address those variants, and we'll have to address them either through vaccine boosters, or I still think there's a huge role for therapeutics in COVID. And I think a lot of the drugs that we're using, like CCR5 antagonists and statins in long COVID, can also help individuals from going on to severe disease and death in acute COVID. And frankly, that's what the vaccines are doing. They're effective in preventing severe disease and death for the most part. And the issue is, I think we can achieve that with therapeutics as well, so that we can attack on multiple fronts the mutation rate and variants of this virus. It's got to be done. And I've talked a lot about using immune modulatory drugs, like I said, the CCR5 antagonists, and ivermectin's been shown to have immune modulatory effects, and obviously the statins have a very important role in preventing the immune and inflammatory components of vascular inflammation. And I think we have to really use these sequence agnostic therapies more readily, because to come up with something that's sequence specific against the virus, you're susceptible to mutation in the virus, developing resistance, much like HIV did.
Wonderful. Dr. Patterson, you have been in many pandemics. COVID-19 ranks among the deadliest viral disease that we have faced as a species. Is there anything that uniquely distinguishes the causative virus, COVID-19, from the viruses responsible for other pandemics? Let's say, as an example, Spanish flu of 1918, smallpox, HIV, replication rate, virulence, infectivity, mutation rate. Do any of these features stand out for COVID-19 and resulting mutants in posing greater challenges than other historical viruses?
Well, again, you just described why this is such a challenge. You have a virus that is very complicated, it has a high mutation rate, and it's got a pathogenic mechanism that is distinct, and like I said, unique in that it causes such profound vascular abnormalities. HIV caused immune suppression abnormalities. So, each of the viruses has their own what we call tropism, or ability to infect certain cells, and we have to look at it pathologically, in terms of what's the outcome of infecting a certain cell type in terms of normal bodily function. And it's very interesting in the early days of acute COVID and acute SARS-CoV-2 infection was that it decreased CD8 T cells. So if you think of an appropriate antiviral response, you have CD4 T cells and CD4-8 T cells that kind of work in partnership to fight off viral infections and frankly other infections and cancers, etc. And in HIV, CD4s were low, and in COVID, CD8s were low. So many of the early patients were supremely immunosuppressed. And that obviously allowed other opportunistic infections to come in, or reactivation of chronic viruses like CMV and herpes and others that infected some of these individuals but lay dormant because they were controlled by the immune response. So again, every virus is different in terms of the cells that it infects, its ability to replicate, its ability to mutate. The hallmark of transmission is the ability to create high viral loads in the specimens that actually are responsible for transmission, whether it be bodily fluids, blood, secretions, droplets from the nasopharynx and the oropharynx in terms of SARS-CoV-2. So each virus is different, each virus is unique, and I think one of the things we have to do going forward is come up with strategies to fight off emerging viral infections that are agnostic to the virus, and that would be by enhancing the immune system, much like the wonder drugs of cancer the past few years, the PD-L1 inhibitors that are broad anti-cancer drugs. They're not targeting cancer genes per se, but what they're doing is they're allowing our own immune system to fight off those cancers in a more efficient way. And I think we have to think about that strategy as it relates to infectious diseases as well, coming up with ways to make our immune system, embellish our immune system and allow it to function better in eliminating these pathogens.
Impressive. Are there any concerns about increasing frequency of outbreaks by newly discovered viruses in future? What we can do to protect ourselves?
Well, like I said, I think it's coming up with strategies of what to do when there is an emerging viral infection. I think that's very much part of it, and then continuing surveillance worldwide for pathogens, that could come from different sources, such as animals and such. So I think it's all about surveillance, and it's all about being prepared with broad spectrum antivirals and antibacterials, for that matter, since we can't just we just can't talk about viruses, but we have to talk about fungi. TB, multi-drug resistant TB is a good example. Bacteria that may be resistant to antibiotics. So I think we do have to come up with a concerted strategy that allows us to address the future in terms of pandemics.
Thank you. What are your perspectives, Dr. Patterson, about the new emerging therapies and new vaccines?
Well, again, I think it's another weapon in our armamentarium, and like I said, I do think therapeutics are going to play a role, and it's a battle. Game's on. The vaccinologists need to battle the virus. The therapeutic folks, whether it be neutralizing monoclonal antibodies or immune modulating monoclonal antibodies, we're in a war, and we have to fight it on multiple fronts. And I don't think we can stop or say one area is better. I think we have to just pursue it on multiple fronts.
Thank you so much. Dr. Patterson, right now some states like California are again putting some measures like masks, considering opening the schools. What are your insights in regard to this subject?
Well, it's a good question. I think we have to consider the demographics, and we have to consider the pockets of emergence of variants in making those types of decisions. And if we have to go back to masks, we have to go back to masks. I mean, it's unfortunate, and I'm not saying that we will have to, wide scale like we did before, but the hallmark of controlling infection is keeping it isolated. And in certain areas with high incidence of infection with variants, absolutely we're going to have to use all possible means to stop the transmission. And that's no different if you're thinking about an Ebola outbreak, in Africa, where the whole key is isolate it, wall it off and keep it from escaping that area of high incidence. So, I think we're at a point where we have some measure of control, and I think we can focus on the areas of variant infections.
Thank you so much. Last not the least, Dr. Patterson, InCellDx is a fascinating company committed to advanced precision medicine. Would you tell us more about your wonderful team and the wonderful research they are doing?
Yeah, it's interesting because some people have called us a research lab, some people have called us a testing lab, but the bottom line is, we're a biotech company, and we focus on precision diagnostics, and more we're getting into therapeutics and therapeutic targets, and using our basic science research and our R&D to not only develop appropriate diagnostics for certain diseases, but to identify new targets and make them part of our company. And I think that puts us kind of in the middle of diagnostics and therapeutics, and that's a good place to be, because I don't think you can have one without the other. Drugs and drug development is great. There's not a one-size-fit-all drug out there, so we have to use precision medicine, which involves using diagnostics to say, before you give a drug to a person, that drug is going to work. Right? Back in the old days, it was kind of like doing an experiment on a patient where, let's try this drug and see if it works. And then a few months from now, we'll know either it worked or it didn't work. As opposed to, the last, 10 years or so, where the major focus has been, let's decide before a patient takes a drug whether or not they're going to respond. So it's predictive medicine, it's precision medicine. You can't have one without the other. It's foolish. And when you design clinical trials, you can't have one without the other. You're not going to give a drug to a patient hoping it's going to work when they're lacking the biomarker that says it's going to work. And that's how medicine needs to be deployed. And I think that's the most efficient way, and it's the fastest way to get help where help is needed.
Impressive. Thank you so much, Dr. Patterson. Again, so many thanks for accepting my invitation. I'm so honored and privileged for this interview. Thank you for your time and the wonderful information. Thank you so much.
It's a pleasure. Thank you.
با سلام خدمت همه بینندگان عزیز تلویزیون جهانی کانال 1. خیلی ممنون که بیننده برنامه ما هستید. همونطور که دیدید مصاحبه امروز ما با دکتر پاترسون از دانشگاه استنفورد، ایشون کاندیدیت هستن برای جایزه نوبل در آینده، پتانسیل کاندیدیت، و کارهای خیلی بزرگی را انجام دادن در زمینه ویروس شناسی و در 4 تا پندمیک ایشون حضور داشتن. اطلاعات خیلی خوبی رو دادن به ما که ما حتما ترجمه ورد بای وردش رو خدمت شما در برنامه‌های آینده خواهیم گفت. برنامه ما هر سه شنبه ساعت 6 بعد از ظهر به وقت غرب آمریکا و معادل با 5:30 صبح به وقت ایران پخش میشه و ما تازه ترین یافته های پزشکی رو در این برنامه به اطلاع شما میرسونیم. تا برنامه بعدی، روز و روزگار خوش.